Hello, my name is Rajpreet, and I'm excited and grateful for the opportunity to share my summer research exploring cholesterol as a key modulator of prion diseases. Prion diseases are 100% strictly fatal neurodegenerative diseases of humans and some other mammals that are caused by the misfolding of the normal cellular prion protein PrPC into the infectious isoform termed scrapie prion protein or PrP-scrapie. This misfolded form accumulates in the brain and contributes to the severe neurological symptoms observed in affected individuals. A unique aspect of these prion diseases is that the infectious protein PrP-scrapie acts as a template which induces further misfolding of normal PrPC into the abnormal form. This is known as a self-propagating cycle of conversion, which allows prions to spread within the brain without needing nucleic acids. To briefly outline the mechanism, the pathogenic protein conversion process of PrPC to PrP-scrapie occurs at the surface of cells, where PrPC is anchored to the plasma membrane via a GPI anchor, which is a lipid molecule. Importantly, this anchor places PrPC within specialized membrane regions known as lipid rafts, which are enriched in cholesterol. Cholesterol helps maintain the structural integrity and organization of lipid rafts, and therefore any changes in cholesterol levels within these raft structures can severely influence the localization, clustering, and function of any membrane-associated protein, including PrPC. Alterations in cholesterol homeostasis have been shown to impact not only prion diseases, but also other neurodegenerative diseases that involve pathogenic protein misfolding, such as Alzheimer's and Parkinson's disease. Using two neuronal prion-susceptible cell lines, N2A and CAD5, we wanted to establish a cholesterol overload model mimicking the elevated membrane cholesterol observed in prion-infected neurons, and we also wanted to investigate how this cholesterol enrichment affects total PrPC expression and PrPC surface localization. To establish the cholesterol overload model, we treated confluent N2A and CAD5 cells with water-soluble cholesterol complexes for 4 hours, and then confirmed cholesterol enrichment using the Amplex Red assay shown here, ensuring that the treatment effectively increased total cellular cholesterol levels. Our experiment was structured using numerous complementary approaches to examine the effects of cholesterol on prion infection. After establishing the cholesterol overload model, Western blotting was performed according to the steps highlighted here, only on CAD5 cells to assess total PrPC expression under both control and cholesterol-enriched conditions. Unfortunately, Western blotting was not conducted on N2A cells due to time restraints. Using N2A cells, we conducted flow cytometry to quantify surface expression of PrPC because usually N2A cells exhibit lower overall PrPC expression compared to CAD5s, so making it important to assess surface expression before any downstream analyses were conducted. For the experiment, we treated the N2A cells with cholesterol and then followed our lab's established flow cytometry protocol as is highlighted here. Here is a figure depicting cholesterol quantification using the Amplex Red assay. We found that cholesterol levels significantly increased in the cholesterol-treated cells compared to their control counterparts, indicating that our cholesterol overload model successfully elevated cholesterol levels, providing a valid system to further investigate prion protein mechanisms. This is a Western blot of both cholesterol-treated and control CAD5 cells. On the left, we see no visual bands of PrPC, indicating that these cells specifically expressed very low or undetectable levels of PrPC under the conditions that were tested. On the right-hand side here, we can see the characteristic 3-banding pattern of PrPC in both the control and cholesterol-treated cells, but no differences are observed within the treatments, indicating that the total PrPC levels were unchanged by cholesterol. This flow cytometry data from one sample of N2A cells is representative of all the samples we worked with that show that surface PrPC levels remain largely unchanged following cholesterol treatment and that the N2A cell viability was not affected, considering that 99% of them are still alive. With this data, our cholesterol overloaded N2A cells can now be used for downstream analyses. In conclusion, we successfully established a cholesterol overload model in both N2A and CAD5 cells. As such, the cholesterol treatment significantly increased total cellular cholesterol levels, indicating that cholesterol was successfully uptaken into the plasma membrane. Additionally, Western blot analyses showed no changes in total PrPC levels following cholesterol treatment, suggesting that cholesterol loading did not impair PrPC expression levels. Future studies, including these shown here, will aim to further assess how cholesterol impacts prion propagation in both infected neuronal cells expressing PrP-scrapie and uninfected cells. I would like to thank Dr. Sabine Gilch and everybody in the Gilch lab, specifically Gokin, and the O'Brien Institute for Public Health for funding this research. Thank you for watching. And these are my references.